Hello, my name is Mike Ward. I'm the Global Director of Content at Informa Pharma Intelligence. We're here in Cologne at the Bio-Europe meeting, and I'm joined by Dirk Reyn, who is the CEO of eTheRNA, which is a newly minted, or a company that was created in 2013. So Dirk, thanks very much for joining us. So I guess the easiest question, or probably the most difficult question, is what was it that was that needed fixing that you guys that you got a solution for in the first instance?
Yeah, so I think we're a company that focuses on activating the immune system, and so I think in the treatment of infectious diseases and cancer, I think people realize that we probably will go to a type of combination therapy where you have products that may influence the microenvironment, you have products that basically slow down the defense systems, and that you also need products to basically activate the immune system. And people have been working on very specific targets, single targets to do and try to activate the immune system. But the unique about the work of the founder, Chris Tielemans, is that he looked at the activation process of dendritic cells as a central cell in the immune system, and that is a three-step process. And so he looked at different mixes of molecules that basically modulate that process, and he found a mix of then three components, and that mix was more powerful than any other combinations. So instead of working on one target, he said if you really activate the immune system, you need to work on the three steps. And that's what we try to fix.
So what you're hitting sort of immunoactivation in three different ways?
In three different ways. So there's a danger signal to basically alert a cell that we influence. Then you have the interaction between the dendritic cells and the T helper cells that we influence. And then ultimately the activation of cytotoxic T cells. And it's each of these three steps that we influence.
So obviously, that's what differentiates you from many other people who are either in infectious disease or in immuno-oncology, as it's now called. Presumably, does this have an impact on how you're able to contain resistance? Because one of the big problems, for example, with antibiotics, and also we see it with a lot of cancer drugs, is that through adaptation, the cells, through mutations or whatever, the cells end up being resistant. So by hitting the three targets, does that improve the system, or?
No, it's a good question, but we hit the three targets and we do that using mRNA.
Right.
So the cell basically takes up the mRNA, then prescribes a protein, and it's the protein that is active. And that protein is active for about 10 to 14 days. Right. So we basically boost the system. It works for 14 days. Our drug is basically gone after a day and a half. It's the body that takes over the function. So we've exposed about 150 patients. We've got really people with melanoma, advanced melanoma, that we had complete remission, and that now after 3 years are still still complete remissions. So we have no indication that this mechanism induces any resistance, because we basically kickstart the body a little bit and then let the body do its work.
Right. Okay. So these approaches, quite a lot of the for example, the cell therapies that we're now seeing are very, very specific to the patient. Yep. Is this something where you can have a more universal approach, or are you still very patient specific?
No, it is a little bit more universal. So we add the mRNA to basically tell the body to basically boost the immune system. We do add the tumor-associated antigens that have identified to point to the reaction a little bit. And we may in the future, if the whole story about specific neoepitopes and antigens proves to be really, really valuable, we have the option to add that to our product. But right now we're more in the clinical trials, basically providing a broad immune stimulus.
Right, so you mentioned that the invention was by who?
Yeah, so the professor who invented it was Chris Tielemans from the University of Brussels. Right. And so he's a well-known mRNA and dendritic cell specialist, and he's got about 150 publications.
And the technology has been licensed to your business? I mean,
yeah.
how did, the company was created in 2013. Who created it, and You know, what is the relationship between the founder, the science, the intellectual property, and eTheRNA?
Yeah. So the company was created as a spin-off of the university, a small, and it was at that time based in the university. With 12 million non-dilutive money, they created a data set in 150 patients to validate the concept. And that was like very intriguing.
So that 12 million euros, that's what, from grants?
From grants, from European projects, from local Belgian projects. But then I think the company was mature enough to grow and then try to get more money. But obviously the team in Brussels was looking for some business people to join. So my background is I used to work at J&J. I was also the founder of Movetis, which we then exited to Shire. So I was looking at different interesting technologies. And with some people, we basically invested in the company, so we became a major shareholder. And then we optimized the development plans and the business plan, and then we're able to raise 24 million in March of this year with investors, major international investors. And now we're taking the project forward in melanoma, in breast cancer. So that's a little bit the story.
So that was the Series A round that you did.
The one in March,
24 million euros.
Euros. Yeah.
So what did you promise the investors that eTheRNA was going to do over the next few years?
Yeah, so the promise is that, and it's building on what we discussed before, the interesting clinical data was created with a cellular product in which you brought the mRNA, the trimix mRNA and the antigens in the cell of the patient and brought it back. But intriguingly, look, if you take the naked mRNA and you inject it in the lymph nodes or directly in the tumor, in a battery of tests, we saw exactly the same results. And so we made a promise to the investors that we're going to develop a very easy to administer mRNA-based product for melanoma. And so, or in monotherapy, so after people have, for instance, their lesions removed, that we can protect people from recurring, or in metastatic disease, we probably then would combine it with checkpoints, and that we have an increase on complete remission rates, we hope in the range of 10 to 15%.
Right. And are you planning to do this on your own? I mean, what other technologies or partners do you need to bring on board to be able to deliver that promise?
Yeah, so right now we're a young company, we're about 25 people, so we have the manufacturing we keep in-house, and because that's the flexibility and the nice thing about mRNA production. But we are looking for, I think we're definitely looking for partners on the formulation side, because we're now injecting intranodally. There are developments that maybe you can develop in different, so formulation partners is something that we would definitely be interested in. And then, I think, as you said, it is a combination market, so I will work with people that have other products, because my product is probably in a lot of settings will have to be combined. So our model is going to be with partnership to basically take it forward, and that will include people that have checkpoints or other interesting compounds, which a combination of our product may be helpful.
Right. Okay. So what's the next big milestone for eTheRNA?
Yeah. So the big milestone is, we're starting the first quarter of next year 2 trials, one in triple negative breast and one in melanoma. There we're using this direct administration approach, and so we'll have readout of that in the second half of 2018, and then we will have basically shown that we have an off-the-shelf product that we can easily administer and that it is effective.
So those trials, that sort of safety, will there be any sort of demonstration?
There will be safety, there will be extensive immune monitoring, there will be extensive looking at tumor environment, everything happens in the tumor. So we're going to try and prove that we can indeed create T cells populations in the tumor that then basically attack the cancer cell. Right. Again, the classical phase called turn cold tumors into hot tumors by basically stimulating the immune system. And if we've proven that, I think we have a wonderful product there.
So what in all this, because obviously it's sort of the approach, one of the things that sort of held back the idea, for example, of immuno-oncology was that idea of turning cold tumors into hot tumors. What's the what's the biggest stumbling block do you think that might be in the development of your project? What are you least looking forward to in terms of the results?
Well, I think I'm not concerned about safety because like I said, we tested the product in 150 patients, so that's it. But okay, I think it is a competitive market, so. Yeah. I think because there will be a lot of combinations, we know that I think in order to become a gold standard in combining with other things, we will have to prove efficacy and show that we're probably at least 10-15% add on current treatment, I think, to be accepted. So I think it's proof that we're effective.
And presumably when you're at that stage, you're going to have other partners to help you with that.
Yeah, the beauty is we stimulate the immune system. We're now focused on melanoma breast. That's a choice we made, but the product should also work in other cancers, should also work probably in infectious diseases. So once we've proven that, I think we're going to try and look for other partners to then further develop in these other indications.
And your 24 million euros, that's going to take you to what stage?
That will take us beyond the stage that we can prove to people that we have an easy to administer effective product, and then depending on different options that we have and the people that are interested will then decide what we do best.
Okay. Dirk, well, thanks very much for turning up
and
sharing the story.
Okay, thanks.
Cheers.